Hello and welcome back, friends. In this video tutorial we'll be talking about diffusion and osmosis. Now this is one of the basic concepts of life science and chemistry, and whatever, this is a fundamental concept that you need to know as a scientist. Now what is diffusion? Diffusion is a situation where the molecules transfer themselves from the high concentration to the low concentrations. Okay, it's a general rule of rule of nature. Like, say, if you take a glass of water and you take a very condensed color, which is also the color is also produced in the water solution. You take that color and you put a drop of that color, let's say the ink, if it's the ink of the marker or a pen, you put that ink into the drop of ink in the water. You will see that ink will slowly mix with the rest of the water of the glass, and it will make the whole glass equally colorful, not the small part of the glass or something like that. This is so solute tends to transfer or tends to move from high concentration to the low concentration. That is the idea of diffusion. Okay, so let's say if I draw the situations like, say, this one, this is a solute. Let's say this is the ink particle that we are talking about, the ink particles. It is present in very high concentration here, and here it is moving. So what will happen? The particle from this high concentration will move to this low concentration of that same particle, obviously. Okay, and when until they reach, or they become the similar concentration, until they reach in the equivalent concentration, and after some time what you will see something like this. This will be known as the equivalent concentration. So they will reach the equilibrium for the diffusion. Okay, this is the idea of diffusion. On the other hand, if you look at osmosis, with the idea of osmosis is also very similar with diffusion, but in this case slightly different. But before going into there, I must talk a little bit more features about diffusion as well. Now the diffusion, as you see, molecules or particles, whatever you choose here, in this case solutes in the basic term, they will move from high concentration to the low concentration. Now this movement always work down the concentration gradient. Down the concentration gradient means from high to low every time. This is the nature, this is the truth, and until and unless they reach the equivalence or the equilibrium. Now what other things that influence this diffusion process? One thing is the temperature. If you apply temperature to this high concentrated situations, if you let's say you just put some molecules there, and if you increase the temperature, you will see the movement of those solute particles will be much faster with increasing temperature. So temperature will help the diffusion process. Okay, the second thing that influence is the pressure. If you increase the pressure inside, the diffusion potential of those particles, it will go slow, and if you release the pressure, they will start moving. Another thing that also regulates this whole process is again the difference in the concentration, obviously. Now let's talk about the osmosis. Okay, now we all know that. Now let's talk about osmosis. Osmosis is also following a similar rule like the diffusion, but this case is little bit special, and the thing that is making the case special is a semi-permeable membrane or a selectively permeable membrane. Now what does that mean? You know, in osmosis the same scenario is working, the same scenario is going on. Let me okay, let's take this same thing. Let's say these are the molecules from, this is the high concentration, okay, and this is the low concentration of that same particles over there, same solute over there, and they are separated by the semi-permeable membrane. So this is the semi-permeable membrane, let's say semi-permeable or selectively permeable membrane. That means this membrane will allow molecules to pass selectively. So this membrane will not allow all the molecules to pass. Now, if you look at a solution is made of two things, a solute and a solvent. Okay. For example, for our understanding sake, let's say in this case, separate this up. In this case, we are talking about glucose water solution. Okay. In glucose water solution, if it's a glucose water solution, then what will happen here? These are the glucose molecules, for example. The glucose molecules are present in high concentration here and low concentration here in this chamber. Two separate chambers separated by a semi-permeable membrane. That membrane could be parchment paper, or let's say it is a cell membrane, because the feature of cell membrane is also similar that of a semi-permeable membrane. So what does that mean? This semi-permeable membrane will not allow glucose to pass. It will only allow water molecules to pass. Now let's say the water molecule, if I draw water molecule, okay, let's draw it with black dots. These are water molecules, for example. Okay, so now try to understand the scenario very clearly. As you see it here, in the second compartment, the concentration of glucose is less. Why? Because there is more water, less glucose present. While in the first chamber, concentration of glucose high. Why? Because less water, I mean the ratio of the glucose to water is high. So that means higher concentrated concentration of the glucose. So now in this case we know that glucose cannot be transferred through this semi-permeable membrane. Only water can transfer. So what will be the idea? According to the idea of diffusion, we know that the molecules will move from high to low concentration. Here, high concentration of glucose here, low here. So we can think that glucose can pass from high to low. But the answer is no, it cannot, due to the semi-permeability of the membrane, where it is written glucose no, water yes. So let's assume this scenario oppositely. The water concentration is high in this chamber but low in this chamber. So water can flow from this second chamber to this top chamber, right? So that means water is flowing from the high water to the low water, and water is flowing from the low glucose concentration to the high glucose concentration. Why? Because water can flow. So if water start to flow, then what will happen? After some time, what we will see is the water molecules, more water molecules there. So what it will do? It will balance the concentration of glucose in both the chambers. Isn't it right? Because as we cannot transfer glucose, let's transfer water in the opposite way. By the same manner, we will make the concentration of glucose in both chambers same, and that is the idea of osmosis, where one particle will not move but the other particle will move, and to maintain the concentration in both the chambers same. So this process of water movement will continue, and it will stop when the concentration of glucose in this chamber and this chamber will become similar, it become very at the equilibrium. Then they will stop. This is the idea of osmosis. Now again, osmosis is also influenced by temperature. High temperature will increase the osmosis. Another thing, that this permeability feature of the membrane also controls the process of osmosis. Now if you punch a hole in the semi-permeable membrane, then it will the whole process will get destabilized. And another thing, it will also depend on the distance between the two chambers. Okay, the distance where the water molecule needs to travel from one to next chamber, that also important. If the distance is more, then the rate of this osmosis will be less. If the distance is less, the rate will be higher. That is the idea of diffusion osmosis. So diffusion and osmosis both are equally very important scenarios from the perspective of life science and cell biology, and for the living of the cell. Why? Because the cell membrane behaves like a semi-permeable membrane, and the cell membrane sometimes receives something or some molecules, let's say it receives oxygen, it receives release carbon dioxide and stuff by the mechanism of diffusion. So if the oxygen concentration is higher outside the cell and the oxygen concentration is low inside the cell, it will take the oxygen from high to low through the membrane. It will not involve in any sort of osmosis, only diffusion, and that's how cell gets the oxygen. On the other hand, sometimes some molecules needs to be transferred across the membrane. For example, I told you about the glucose solution and stuff, where they needs osmosis to properly uptake glucose and other molecules, or to make everything is balanced. Because if they are present in the imbalanced situations, there will be change in the osmolarity in the cell. So the cell could die, cell could burst, or cell could ultimately shrink in those conditions. What are those conditions and what are the situations where the cell needs to regulate its osmolarity? We will talk in the next video. We'll talk about the osmolarity, we'll talk about the tonicity of the medium where we put the cell, and the different types of situations regarding that in the next video. But that is the conclusion of diffusion and osmosis. If you like this video, please hit the like button, subscribe to my channel to get more videos like that, and share this video with your friends. Thank you.